Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program. Local broadcast is made possible in part by Pfizer.
The topics that we will talk about today is including what are we talking about terrorism? The bio, I think you all got pretty much coming in the door. The terrorism really is talking about the way people feel and about the sociology and dealing with fear. So we'll start off with fear. And then we'll go over some of the most suspected agents. And these aren't guesses, and really it's not just about the medicine of the natural infectious diseases, but there's a history of proof of principle and actually nation states, including our own nation state, the United States, actually using good scientists, good engineers, good biologists, in a formal institutional setting to actually build weapons up to the point where they were deployable. I'll present a theory for you, and it's my video store theory of fear and bioterrorism. Now, I'm not talking about the shows like 24 or any of the number of sci-fi movies of outbreaks, per se. I find two predominant subgenre of the horror genre films. I find unstoppable forces of nature, and they fall into the category of monster flicks. And of course, germs are unstoppable forces of nature. We feel that way, and we do things to stop them. But for most of human history, they were sort of out of our control. People got sick and they died. People still get sick from germs, and we still die of infectious diseases. And then the other subgenre is unrepentant evil, and these fall in the category of slasher flicks. And so when you talk about bioterrorism, we have people. It's perceived we have people, maybe countries, that are willing to use germs and use them to hurt us. So we cross these, we cross, and maybe at our core, there's some sort of basic repulsion or basic elements of our human nature that we don't like unstoppable forces of nature. We like to be in control, at least to adapt to nature. And we don't like unstoppable forces of evil either. Coming out and trying to do us harm. What is bioterrorism? Pretty clearly defined these days as the malevolent use of bacteria, viruses, or natural toxins, bacterial toxins, against humans, and we'll think mostly about humans from this point on, but also the animals and plants that we rely on for our well-being. There's worries of bioterrorism against our livestock and against our crops, as well, hoof and mouth disease, plant pathogens that would cause untold troubles if they got released. An attempt to cause harm, that's direct physical infections, and mainly to create fear. There's many ways to kill people, I'm sure, and this may not, the use of bioagents is probably not the most efficient way, but because of the fear element, it's believed that it is one of our nation's most vulnerable points, both militarily and civilian populations, to cause disruption in our society and screw around with our minds and our health. And this list includes bacteria, which are free living. Some examples of diseases are plague, anthrax, tularemia, Q fever. The viruses, smallpox, Marburg, Ebola, Lassa, dengue, monkeypox. Products made by bacteria, botulinum toxin, tetrodotoxin, ricin, it's actually a plant toxin, and genetically engineered microbes, mixing Ebola with influenza or something that may or may not exist at this point in time, but there's worry that people with some technical expertise could start genetically mixing the genomes of these different viruses to make something new that could infect us. All right, is this a modern, with all this technology and the use of germs and all what's been written in the fiction categories of human imagination, is this a new problem? But why would anybody want to in the first place? Why use germs to hurt other people? Well, of course, they're small, and they're undetectable by our five senses. We can't know if we've been exposed to an infectious disease until we get sick, by and large. They're relatively inexpensive to produce, to develop, and to deploy. They're 600 times less expensive than chemical weapons, which you'll hear about next week, I believe, and about 800 times less expensive than nuclear weapons. They're relatively low tech to make and deliver, and anyone that can bake a souffle probably can follow directions enough to grow microbes and weaponize them, at least to a small degree. They're effective in small amounts. You could be carrying them in your pocket, and they're hard to trace. There's no signature to current technologies to know out of a background of all the organic life forms and all the bacteria and all the viruses in the world, which ones are the dangerous ones. It's a challenge to people building detectors to identify the ones that could be released on purpose from what is normally out there in the world. The delay in symptoms, you could become exposed, and there's an incubation period, allows for anonymity of the person releasing them. So a person could put spores into an envelope, or release spores or germs into a shopping mall or into a building or into the big house, and people would go home, and it wouldn't be until 10 to 14 days later, depending on the agent, that people would start getting sick, and by that time a perpetrator could be long gone and very hard to trace. There is the chance of mass casualties, and I'll show examples of that, not just individual people getting sick, especially with the diseases that are contagious that can be passed from person to person, and you start getting an amplification effect. One person gets sick and infects several others, and the numbers grow geometrically. We are afraid at some level, most of us, of getting sick. We don't like it at all. And it causes a ripple effect with people's behavior that are the victims and people that think they may be the victims of a biological agent. And there really is, it was pointed out in 2001 on the heels of 9/11 with the postal anthrax attacks, that there's a lot of panic and disruption. And it's very expensive to treat people, or test people even, and to treat them if they are actually sick, and then to go around and decontaminate the places where there still may be harboring infectious agents remaining. The germs can be put in our food supply and our water supply. This is a little less likely. The scenarios are predicted not that water attacks aren't so well. But you know, there's been papers on the vulnerability of our milk supply and different food supplies. But mainly the one we're worried about is germs in our air supply. And people putting viruses and bacteria and botulinum toxin and other toxins in the air. Now, it doesn't have to be as dramatic as a crop dusting plane. It can be vents. It could be a number of things. The possibilities are limited only by imagination. One scenario that was not predicted was germs put into our mail. And there's a zillion other ways and means that we can think of to spread these things about. Now, on top of that, some diseases are transmitted. They are contagious from person to person, like smallpox and plague and Ebola. Some are very contagious, like smallpox, plague, and Ebola. And others are not. And sometimes I don't know if lucky is the right word, fortunate is the right word, but in the 2001 anthrax attacks, you had to be directly exposed to get sick. In the 1400s, the Tartars were laying siege to the Black Sea city of Kaffa, and they could not get past the city walls. And one thing they tried was to take people outside the walls that have died of plague and catapult them over the walls in the hopes of starting a plague epidemic. Now, this was not a very good strategy. You know, of course, you're dealing with plague, and you're likely to get it yourself, and the city fell. And plague was the reason. Now, what I didn't point out, so there was motivation here, and they had sort of a vague idea that this might happen, and plague did happen. But of course, as we'll come to, there's probably the dead bodies being flung over the walls was not what caused the start of the epidemic of plague inside Kaffa. It was probably the rats that just go freely through the walls. Before the Revolution, Sir Geoffrey Amherst was commanding the British troops in America, and they were having troubles with the Indians, with the Native Americans. And he sanctioned the use of blankets that were used by smallpox victims as germ warfare agents against the Indians, who had not seen smallpox before, and they were very susceptible to the disease, and it was really a form of genocide. In Japan, in Tokyo, the Shinrikyo, a Japanese cult, here was their leader, attempted several unsuccessful biological attacks in Tokyo with both anthrax and botulinum toxin, and they were unsuccessful. In the instance of their anthrax spore attacks, they were up a bridge above a road that led to a U.S. military base, and they were spreading spores around. And they did have some expertise on their staff. They were actively recruited biochemists and physicists and biologists and law enforcement agents, and they were even trying to buy off politicians. And their success in terms of anthrax was a failure. There was no success because they got one thing wrong. They were using the vaccine strain instead of the hot strain of the disease. So they were close, but ultimately didn't get it quite right at the very first principles. In 1984, there was a cult in Oregon, and it's the Rajneeshis, and there's their leader. And it was a religious cult in Oregon, and there was an election, and they wanted to, there was a vote for them. I forget if it was zoning or they wanted permission to build a compound. And and the locals didn't want it. The people in this town didn't want it. And so they wanted everyone not to show up and vote the next day. So they went around and put the food poisoning, it's a rather mild food poisoning organism, Salmonella typhimurium, in the salad bars at all the restaurants in town. And 700 people got sick. I guess in this town in Oregon, the tradition is go out to dinner the night before the election or something, I don't know. And 700 people got sick. And they lost their vote, by the way. Even though they were sick, people take their civic duty. You know, you can run from the booth to the restroom real quick, I guess. But they lost their election. And actually, these guys were not caught. No one really understood that it was actually a bio attack in the salads. What happened was one of the people in the cult turned them in years later and confessed to, and then years later they were arrested. It was a cold confession. No one was even aware that it was a bio attack and not a normal food poisoning outbreak. The one that changed my world was on the heels of 9/11 and the anthrax attacks, when people put spores in a couple of envelopes and mailed them around to various members of Senate, to news media, Tom Brokaw, down to the Inquirer. People started getting sick in October. There was 22 cases of people actually getting sick. 11 were the milder skin form, cutaneous anthrax. 11 were the more very serious and cases of inhalation anthrax, systemic anthrax. And there were 5 of those 11 people died, which is less than half, still a lot of people, less than half, where in the case where systemic anthrax, until that point in time, was considered to have 90 to 95% fatality. Now, there's been so few cases of inhalation anthrax in the modern era that I think the improvements in the ICU saved a higher percentage of people than previously in the history of people getting anthrax. And of course, these few letters and these few cases shut down the government in Washington, and the Hart Building was closed. There was a number of postal system was closed or severely curtailed. On top of this, over 20,000 people were in the same vicinity, in the same area as the infected people. And all these people went in for testing and drugs and antibiotics, ciprofloxacin, take their Cipro for a number of weeks. And a lot of the people that complied with the full regimen of Cipro got sick from long-term use of this very potent antibiotic. So they were small events, but there is a history of potential for big events in biowarfare. And this is nation states. So led by the United States, England, Soviet Union, and Canada had a program of two, starting between the world wars, we had very large, highly developed, technologically advanced, weaponized, and deployable offensive bioagent programs in this century. Under President Nixon in 1969, the U.S. discontinued all its offensive chem-bio programs, and that holds today. In my travels, I have absolutely no hint, no feeling that there is any offensive biowarfare, offensive chemical warfare programs left in this country. We've held fast. President Nixon was convinced by his scientific advisors, including Matt Messelson at Harvard, just predating 1969, that we should stop this. And it wasn't because of ethical reasons, or the use of germs was distasteful or beneath us, or the use of chemicals was distasteful or beneath us, but very practical reasons. He thought, he was convinced, Messelson convinced Nixon that, that we don't want to propagate this sort of technology in countries without our resources. We don't want to give easy weapons that we'd be vulnerable to, or build the technologies or the infrastructures or the know-how to make these weapons to countries or people that couldn't formulate a conventional military, couldn't formulate a conventional nuclear force. We'd be our biggest detriments if we got this sort of weapons to countries and make them a viable threat when they couldn't do it otherwise. A global ban treaty was signed by greater than 100 nations in 1975, including all the ones that were considered potential threats. And the Soviets, they cheated big on this until the collapse of the Soviet Union in 1991. And they kept going, and their offensive biowarfare program grew and grew and got better and better. And still, nations are believed to have active biological secret programs and or stockpiles of bioweapons. One of the offshoots is tracking the weaponers themselves, the engineers, the biologists, the crossover scientists that can make weapons deployable. And tracking these people around the world is important, and these guys are all out of work. And the U.S. has tried to hire them on and track them, but a lot of them can't be found, and they don't have jobs, and we don't know where they are, and we hope they don't end up any place in any countries with any groups that dislike us now. And you can see that there's reserve mobilization facilities, biological weapons production facilities, the research facilities, the testing grounds, storage facilities, and it was very well organized, well funded, and not a shoestring operation at all. Here's a picture of one such facility. It's in Kazakhstan. It's a production facility, and it looks kind of like where I work over there at the medical center. I mean, it tells you something. You work in like 1960 Soviet architecture, you know, it's the same sort of building. And it's about the same, you know, it's on the same scale. And, you know, this wasn't a research facility. This is a production facility. This entire buildings were used to large-scale manufacturing, milling, drying, and processing, weaponization of anthrax spores. If you look at this little picture, this little hill in the foreground with these vents coming up, that was just where they stored the 55-gallon drums, oil drums, full of anthrax spores. And this was a protected bunker that was supposed to be able to survive a reasonably close nuclear blast. And the I guess the premise was that you don't want to let whole-scale nuclear war interfere with your ability to kill even more people. These are all fermenters. There's a person there. I hope you can see it. These are all fermenters that were just making anthrax spores 24/7. They have these aerosol test chambers where you could test the particle size. You can test the efficiency of your weaponization process, and by putting large animals in there. That could hold a cow. All right, in 1979, a city of 1 million called Sverdlovsk, there was an anthrax outbreak in the Soviet Union. Now, Sverdlovsk is a city also renamed after the fall at Ekaterinburg that is just east of the Ural Mountains in Russia now. And it resulted in scores of cases, and 67 people died. And this was reported at the time, and it was claimed by the the Soviets at the time to be a natural outbreak of diseased animals being eaten by people. 67 people ate a diseased cow or diseased meat. And it wasn't until after the fall of the Soviet Union in the 90s and openness and perestroika and glasnost that the pathologists of the hospitals and the medical facilities got to come forth and tell the true story that this was not gastrointestinal anthrax, but inhalation anthrax. And what happened was, if you look at this sort of cartoon map, there's all the populated areas are in gray. This is Sverdlovsk. It's a city about 1 million, so you get a scale. Right there at the apex of the cases, these ovals represent where there were cases or spores were found, was a military vaccine, vaccine in quotations, a military vaccine biowarfare research center. So it was making vaccines against anthrax, or so the claim was. Now, if anything, the Soviets had very good weather reporting system, and it was only a certain day that the wind was blowing in this southeastern direction. And all the cases were in this vector, people that were in this vector. Luckily, this facility on this day wasn't located over here, or far more people would have been exposed. But what we learned from this, and added credibility to our concerns, was that after the news came out, the true news, was that during the production of these of anthrax, somebody forgot to put the biological HEPA filters into the hoods, and a small amount of spores went up the hood and into the atmosphere. And what was also of concern was all the people that got sick were within 3 or 4 kilometers of the facility at the edge of the city. Luckily, this is a pretty unpopulated area. But some of the animals that died were as far as 75 to 100 kilometers from the city, and the spores drifted and they were found in the soil up to 250 to 300 kilometers from the city. So upon weaponization, these things are aerosolized, stay airborne at the right size that reach the recesses of your lungs, unit size about 2 micron spheres, and can travel a long distance and stay airborne for a long period of time. And you can imagine such a scenario where even a small amount of spores released in a highly and densely populated urban area can really reach a lot of people. And the pathology reports came out, and it wasn't gastrointestinal anthrax, but the systemic anthrax through the inhalation route that ends up disseminating throughout the body. And this is just a picture of Cardinal's cap of the hemorrhaging, the red and black, mostly red Cardinal's cap seen in people. This is not so common, very rare if it's GI anthrax, but very common if it is inhalation or systemic anthrax. We'll talk about smallpox first. These are orthopox viruses. There's 2 flavors, variola major and variola minor, and we're the only natural reservoir. Unlike plague or anthrax or many of the other organisms, smallpox is uniquely adapted to people. You can get rid of it in people, you get rid of it in the world. And indeed, that's just what we did. Before modern medicine, it was endemic in people of all lands, and upon vaccination, it was fully eradicated in the human population in 1977. The last case was in 1977, and now we don't vaccinate it anymore. The only stockpiles that are official are one in freezers, in labs. There's one in the CDC down in Atlanta, and one in the Soviet Union. And there's been calls, of course, over the past, since 1977, to destroy those stocks. Neither country has done so. So it was a success. It was probably medicine's greatest success, the eradication of smallpox from people. This is a highly contagious disease, and it's spread in aerosol droplets when people cough, from bodily fluids, tears, and blood. The bedclothes of people that have their scabs falling off. These things, scabs stay viable for weeks. This is what was in the, is what Amherst gave, or allowed to be given to the Native Americans. Now there's a 12 to 14 day incubation period. And there's some good news here is that in modern medicine, if you think you've been exposed to smallpox, you can go in for a vaccine up to 10 days. Well, you can go in up to 7 days, and the vaccine still has enough time to stimulate your protective immune response. You can go up to 10 days and lessen the disease and survive. At about 12 to 14 days, you start feeling sick. There's fever, you feel really dragged out, you start vomiting, you have head and back aches. And you're contagious when you get the fever. So before the fever, you're not contagious. After you get the fever, you are contagious, you're viremic, and can spread it to your family, friends, and healthcare workers, people you meet. And this is, after this, then the pustules break out, the pox. And it's a pustular rash, it's painful, and you saw some pictures, I'll show a couple more. And there's a 30 to 40% mortality rate. And so I would feel it would be a true crime against humanity if the world had to deal with smallpox ever again. So once you start getting the blisters, this is what it looks like. And you saw some even more severe cases. And if doctors see this, they have to be careful now. You just don't think it's smallpox if you come in with this, because this looks a lot like chickenpox as well. Now, chickenpox is a herpes virus, not an orthopox virus. And you don't have to worry if you or your child comes home with with something like this, because it probably is chickenpox, which is very common. They're both very contagious. Smallpox, you see it mainly on the extremities, lower legs, hands and feet, and the face and in the mouth, and less on the body. And chickenpox, even though the location of the pox do overlap some, start mainly are higher density in the trunk, less in the face, and the further distal you go from the center of the body, the less you see. And there is the vaccine. Jenner made the first vaccine with the milkmaids and the cow scabs from a related orthopox, the cowpox. And this still works. And there are still plenty of doses around, not enough for everyone in the country. Healthcare workers and military people and lab workers that work with other pox viruses can get this. It's nice long immunity. You dip it into these made from these cow scabs originally. We believe that this may confer long-term immunity, more than the 10 to 15 years that is written up in the medical literature. But what they do is they scratch you a little bit. Sometimes they scratch you on the butt, sometimes they scratch you on the arm with this little sharp fork, and it doesn't hurt that badly, I don't think. I actually haven't had it. And you get a little blister, and then you're immune. Now the trouble with this vaccine for large-scale use is that that some people still react badly to it, and there are very slight chance of death or serious side effects, especially with the immunocompromised people in our society. And so it's really thought that if everyone in the country, of our 300 million people, received the smallpox vaccine, there'd be a couple thousand people, 2,000 to 3,000 people that might actually die or get serious side effects, life-threatening side effects from the cowpox virus. Botulinum toxin. This is not an infection. It's a bacterial product that's made from Clostridium botulinum. It's a normal soil organism, causes botulism in ill-prepared foods. If this organism gets into your canning process or your sausage, it makes a toxin, and then it dies. So there's no infectious agent in your canned tomatoes or your sausage. And then if you go and eat that food without cooking it thoroughly and destroying the toxin, this protein toxin, it can be ingested, taken up through your intestines, and cause you to undergo botulism or flaccid paralysis. And it's the most toxic substance on Earth. And less than a tenth of a microgram is a lethal dose for a person. It's been shown by the Soviets in the US that it's weaponizable and aerosolizable, so you don't have to ingest it. You can breathe it in, it can enter your body through your lungs, or you can have your food supply tainted. It doesn't survive urban chlorination of water, so people don't believe it can be put in the water very easily. And it's also called Botox, and if you use a little bit of Botox, far less than a thousandth of a lethal dose, it can be used medically as a cosmetic enhancement, which is kind of a terrorism in a different sense. But it's a secreted protein neurotoxin that causes flaccid paralysis. And death is due to asphyxiation because your muscles, the nerves that control your diaphragm don't work, and unless you're put on a respirator, you can't breathe. So again, it's taken up into your body through the stomach or the linings of your lungs and circulated around through your blood system until it reaches receptors on neurons. And these are periphery neurons, the ones that control the contraction of your muscles. So this is where the nerve meets the muscles. And it's taken up by cells, by the neurons. And what it does is it's a protease. So it gets into the cytoplasm of these cells, and it clips the docking and receptor and fusion proteins, the machinery that allows for the release of neurotransmitters into this neuromuscular junction. And the acetylcholine that is released when these vesicles fuse and dump their neurotransmitters into the neuromuscular junction. Now, if you don't get these neurotransmitters released and binding and activating the muscles don't work. You go flaccid and you can't breathe. Now, there's another toxin that you've all probably heard of, tetanus toxins from Clostridium tetani, that works on a different set of neurons. And what it does, it works the same way enzymatically, but it stops the release of the neurotransmitter release from inhibitory neurons, ones that turn off your nervous system. And so all your neurons downstream are always firing, and you get a spastic paralysis or lockjaw. All your muscles are really firing all the time. And these muscular contractions are strong enough to break bones. I think it's perfectly feasible that researchers will, I mean, it's not that hard of a stretch to make a toxoid vaccine like tetanus toxin. For botulinum toxin, there's a number of different serotypes of botulinum toxin, so it would have to be a cocktail of proteins that go into this vaccine. And the AMA, the American Medical Association, is not supporting it because anyone that's vaccinated with antitoxin no longer can get Botox treatment because it wouldn't work. But Botox would work. Botox works the same way. They just put a little bit at the very site, and that deadens the ability of the neurons to cause muscle contractions, mostly in the face, and those muscles go lax, and then the wrinkles fill out because the muscles are no longer contracted. It lasts several weeks, even at small doses, and people have to be on assisted breathing for those times. And what is called into question is our ability to, in a case of mass casualties, get enough respirators in ICU to serve anyone that's a victim. And it brings up societal questions such as triage. Who do we help, and who do we let go? We'll look at a couple more. We'll look at plague. Plague was a scourge. It's called the Black Death. And this disease by itself killed a quarter of Europe in the 14th century, approximately 30 million people. Up until the wide use of antibiotics and good public health, it killed 10 million people in India in the 19th and 20th, or first half of the 20th century. It's still around, it's still endemic. It's got reservoirs outside of people. The bacterium is called Yersinia pestis. Symptoms are general malaise, you get a high fever, pain and tenderness in the lymph nodes, or buboes for bubonic plague, convulsions, shock, and then you get this diffuse changes in your skin color due to lack of oxygen and blood hydrolysis, and turns your skin dark. It's called Black Death. It was written up during the outbreaks in Europe. It became a sort of mixed in with theology and punishment of human beings. There's two forms, a bubonic plague and pneumonic plague. And here's just a picture of the plague organism. Here's a picture of it in blood smears along with your blood cells. And it has a safety pin appearance upon staining, easily diagnosed. Here's some swollen buboes on a leg. And here's some of the blackness that you see. It's the Black Death in the fingertips of a plague victim. It's spread by these creatures. This is its actual size. Do you know what this is? That's a flea. A flea that just had a blood meal. What do fleas eat? They eat blood. They live on animals and they eat blood. And what the plague does is it causes the blood meal to clot and not provide nutrients to the flea. The flea is always hungry. It goes around biting more and more and more. And of course, if its nice warm host dies, its pica or its prairie dog or its marmot dies, its rat, its home rat. You know, it may be a rat to you and me, but it's this guy's castle. It needs a new place to live, and it will, when people come in contact with animals, it will make the jump to people and start biting us. There's very well described ecological and natural life cycle. There's the wild animals, the sylvatic cycle, and it goes from pica or prairie dog in North America to prairie dog, mostly by the flea intermediate, and maybe the germ can live in the soil for a while. That's rare or theoretical, and it goes from animal to animal, and it causes death of these animals. Sometimes the flea or direct contact between urban animals, domestic animals like rats, either via the flea vector or direct contact, it gets into the urban or the domestic rats and rodents, and it goes flea to animal in that sort of life cycle circle. People can get it mainly from fleas, either from even from rodents that live in our houses, or fleas from rodents that people that are outside, or it's believed that direct contact, the germ can jump directly from the animal to the people for people that hunt and work with animals. And you get the bubonic plague. Now, if that bubonic plague goes to the lungs and you start breathing it out, you can get person-to-person spread, and that's called pneumonic plague, and that's very contagious. After the flea bite draws blood, it has to draw blood, inoculates your bloodstream with this Yersinia pestis bacteria. It spreads to your lymph nodes, and then systemically, meaning throughout your body. And then you get the buboes, the swelling lymph nodes, and you get the black hemorrhagic lymph nodes and maybe your extremities. It goes to all your different organs, even through your brain and your lymph and your lymph nodes and your limbs and your livers and your hearts and your kidneys and everything, and you die. But if it goes to your lungs before you die and you start breathing it out, other people can breathe it in directly, and you can forego these earlier steps. You don't need a flea bite, and it's pneumonic plague. So it enters in, you breathe it in, you get pneumonia, and it's usually, if you get pneumonic plague, it's usually 100% mortality if you're not treated. And even if you are treated, prognosis is pretty poor. And then it's the highly contagious form is breathed out. And not only is that far more rapid, many people are at death's door before they realize that this is actually what they have. And the trouble with biowarfare is it skips all these. Now, all these steps, and they just put it in the air, and you breathe it in, and you get pneumonic plague straight off. And so if you see a large cases of an outbreak of pneumonic plague without bubonic plague, then it's a landmark that it really might be a bioterrorist attack as opposed to a natural phenomenon. We'll go back to something that we know firsthand in our lab, and that's anthrax. And that's caused by the bacterium Bacillus anthracis. You know, we talk about bioterrorism, and we all don't like it very much, and we're kind of disgusted with the whole idea. But the study of these germs really has been a huge benefit to mankind. And I'd hate to see our baby thrown out with the bathwater and us not be allowed to fully research anthrax and these other organisms, because we've learned a lot. It was the first germ proved by modern science. And a guy named Robert Koch, a German physician, used it to postulate and prove that these newly discovered microbes that you needed microscopes to see were actually the causative agents of infectious disease. It is a proof of the germ theory that microbes cause disease. Pasteur, who invented pasteurization of milk and worked with the wine industry and fermentations and helped prove that germs could do a lot of things, are really living organisms, used anthrax to show that you could modify the living organism by growing it at a higher temperature, then putting it in animals, and it protected those animals from a challenge with the wild-type organism, or a hot organism. And they did this, was really the first vaccinations, first bacterial vaccinations. Mechnikov was a Russian immunologist, and he used anthrax in his studies to show that the white blood cells in our body were actually not just part of the problem, weren't just pus during infections, but actually were part of our immune system. He's the father of cellular immunology and a great teacher. He looks like a very severe fellow, but he was one kick-butt teacher. Here's a picture of anthrax. This is a hand drawing. And remember, this was before tissue culture and before we could work with mammalian cells very well. And he used to use clear tissues of live animals tacked to his microscope stage and stick these bugs in there. He's the one that defined and observed first white blood cells leaving the vasculature, that white blood cells could actually eat microbes and that they could actually kill microorganisms. So he's done a lot. And anthrax was his model because that's what Koch and Pasteur were using. Also because it's a little bit longer, a little bit larger than most bacteria. It's easier to see using the optics of the microscopes of the time. It's a spore-forming bacillus. Here's a gram stain of it. Identified by Koch in 1876 and used to formulate his postulates that germs cause disease. And it is the causative agent of anthrax. Until recently, it came from handling or eating contaminated or animal meat products, and it's very rare. But it's everywhere that there are grazing herbivores. Here is a friend of mine, Pam Coker of LSU. This is a bison up in Canada, and she tracks anthrax in nature, collects their spores. In this country, it's endemic in the national parks of anthrax, in the deer population of southwest Texas, and through the cattle belts of this country. We'll never get rid of it out of the world. And the way you get sick is the spores of this bacteria can enter the body three ways. Through the skin, which causes most of the time not a very serious disease called cutaneous anthrax. If you eat contaminated meats, and people in very poor countries whose animals die have really little choice but to eat those animals, and they don't always check them out. Or if, very rarely in nature, if the spores get aerosolized and go into the recesses of your lungs. Except for the skin form, and even the skin form occasionally can go systemic or throughout the body. What happens next is the spores are taken up by the same cells that are supposed to be defending us. And in the lungs, these cells act as a taxi cab to deliver the spores inside the body. So with inhalation anthrax or pulmonary anthrax, you actually don't get pneumonia. You don't get a lung infection. That's just where it enters. It happens deeper inside. During the transport from the lungs to the lymph nodes, these spores germinate, they hatch to form the active bacilli that makes toxins and capsules and other virulence factors. They spread around the body to such high titers, such high numbers, that you can get 10 to the 8th, a very big number, per milliliter, per cc of blood, and you're still walking. And then they spread even further. You get to the brain, and you die of shock and death in a few days after symptoms appear. It looks as ugly as plague. You see the blackness of it again. It's, anthracos is a Greek word meaning coal, root word of anthracite. And it's this color of the skin infection that gave the name to the disease in the organism. This looks highly inflamed. It's a smoothed edge, and it's painless. And what happens with these is not as dangerous as it looks. So these things, first of all, don't hurt. And even if you don't receive treatment, 95% of the case will be cured spontaneously just by our own immune system. Now, if you get antibiotics, 100% of the cases can be cured. If you don't get antibiotics, you start poking around in there, sometimes the infection can go systemic. And it heals without scarring, so there is no scars, unlike smallpox or sometimes chickenpox. 11 cases, including a little baby in 2001 of cutaneous anthrax. The progression of systemic anthrax, after you breathe it in, is a little bit worse. And you see a timeline up here in days, and this is sort of an overall graph of basic health with good health and dead down at the bottom. But and there's a couple things I just want to point out with this graph is that the time zero is actually the time after the spores are taken up by your phagocytes and delivered to your lymph nodes. Now, unfortunately, spores can, if they don't meet these phagocytes in the lungs right off, can linger there for a very long time, up to 100 days. So you have to be on ciprofloxacin for a long time because the clock doesn't start ticking until time zero. And the spores that are lying dormant in your lungs are resistant to all antibiotics. It's only the growing bacteria that antibiotics can kill. So time zero really starts upon entry into the body deep. And you don't even feel that bad for the first few days. And then you even feel better for a day, and then all of a sudden you go into a deep shock, hypotension. Blood leaves the fluids leave your vasculature, your blood pressure drops, bottoms out, and you die. Now, in many infectious diseases that end up like this, even with shock, it's a slower ramping, and this is a rather sudden death. It's called sudden death. Now, cattle, which often get it, don't complain of headaches or that they feel bad. A farmer can come out and they look fine one day, and the whole herd can be dead the next morning. The spores themselves don't take up dye, but once they come in contact, these purpley pink things are the macrophages, they're the big eaters, the phagocytes that I put in culture. They hatch out within very rapidly. They have exquisite sensing that they're in the right place at the right time to hatch. After about 2 hours, they've actually killed these cells, and you can see these chains of bacteria leaving the cells and escaping. And they start growing very rapidly and into your blood, in this case, into the tissue culture media. And then you're still walking, you still feel like you have the flu, but this is actually a picture of a rabbit blood vessel, a venial, and you can see some blood cells down here. And this rabbit is still eating, and it still looks healthy by just looking at it, but you can see how many bacteria, these hot dog-shaped things, are clogging up its vasculature. And these things are making toxins, and if it reaches this stage, there's no, you're at a point of no return. Diagnosis of anthrax is hard because the initial symptoms, when you can be cured pretty easily, match a lot of other common diseases that aren't always lethal. The virulence factors are encoded on genes, on the chromosome, and they have 2 extra plasmids that are required in hot strains. One produces toxins, lethal toxin and edema toxin. The other produces a polypeptide capsule, kind of like a force field that protects it from our blood serum immunity proteins. The life cycle of this organism is such that it only grows, we believe, inside animals. If it's not inside an animal, it stays as a dormant spore. Now, this is not like a fern spore or a fungal spore. It's not a reproductive body. At most, one bacteria can make one spore. So what happens is that when the bacteria are growing, if they run out of food and they starve to death, starting to starve, they undergo different morphogenesis, and instead of one bacteria making 2 bacteria, as in normal bacterial life cycle, it just makes one spore. And this spore can stay around for decades. People have pulled out spores of related bacilli from amber, from prehistoric times, and they can still hatch. Once it gets in the body, this is what I showed you pictures of, this breaking of dormancy, this germination process. It's armored up like sort of like a golf ball. And there's big layers of cell wall here. There's big layers of protein putting down, making it very resistant to things that would kill normal cells. So it can be boiled. It's resistant to UV light, different forms of radiation. It's resistant to alcohols and chemicals, and it's not killed by antibiotics. There's very little water in this cell. that protects its macromolecules. And all its genome, all its DNA, all its ribosomes are packed tightly into a crystalline lattice of a chelated chemical called dipicolinic acid and calcium. It's very tough to kill these things. Bleach will work on tabletops. Bleach, 10% bleach. With the spores, yes ma'am. The core where all its cytoplasm, its DNA would be, its ribosomes. Everything's quiet, there's no enzymology, no energy, no ATP. Thick cell wall, thicker than most bacteria, and all these spore coats that go down around to protect it from different things. And this anthrax has also this extra structure called the exosporangium, which we don't think does anything medically. It may be involved in sticking to plants and part of its ecology. We know its DNA structure, its unique DNA structure. We know it has 5,508 genes on the chromosome, and on those two plasmids, more genes, so about 6,000 genes. And the question for us in the lab is which of these genes encodes the next vaccine or drug target or diagnostic target? By looking at gene expression, which of these genes are expressed in the context of models of infectious disease. We can do it by measuring its messenger RNA, or to determine which proteins are actually formulated. The enzymes, the building blocks, and the actual machines that the bacteria use are made during the infectious models of infectious anthrax that we use. One of the things we do is look at which genes of those 6,000 are actually turned on. Green is turned on, red is off. So time goes across this, the first 5 hours of bacterial infection. And each different line across, which is each going up and down, is a different gene of the genes. So some are not turned on at all, or some are turned on all the time in culture. And we want to know the ones that are turned on during an infection. And we can organize the genes in terms of when they're turned on from 0 to 5 hours during the life cycle of the bacteria. And we can see that there's waves of gene expression at different stages of the bacterial disease. And then we can go from there and use our molecular biology and our microbiology and pick from these candidates ones that we think are important. Also bioinformatics, using similar genes from other disease-causing organisms, make mutations and test to see if we knock out one of these genes, whether this anthrax mutant strain is attenuated or does not cause disease. And that we can define as a reasonable target for countermeasures. We can take the anthrax spore, we can isolate all the proteins, and we can chop it up with enzymes or chemicals, and we can determine the sequence, the fingerprint, the nature of each individual protein using a technique called tandem mass spectrometry. We get their spectrum, their mass charge ratios, we learn their amino acid sequence, and we go through our databases and convert the spectra into direct peptide sequences, and we can see and identify the proteins that are actually present during any stage of the infectious cycle that we window. And we get pretty good data. This is the only data slide in this talk. The proteins that we see are a subset of all the theoretical proteins from the genome sequence that we know. So it's good non-bias from these techniques. And the actual, there's no large classes of proteins that we can't identify, either extremes of charge or of molecular weight. It's a good spattering, and we get a limited subset of the potential proteins, and there we can home in on as potential countermeasure targets. The vegetative bacilli is what's growing in your body and making you sick. They grow very rapidly in your body. They double every 15 to 20 minutes. But these, at least in nature, are non-infectious. So this doesn't get if you come across this outside your body, it's not going to make you sick. It's the spore which makes you sick. It's stable for decades. It's resistant to anything that we could put in our bodies to kill it. These resistance properties have also made it feasible to be deployed in sort of bombs that would destroy most living things because of the heat. So they can be deployed by bombs as well as sprayed or whatever. And yet at the same time, they're very sensitive to recognize that they're in the human body, and they can germinate within minutes of contact with our macrophages. They don't germinate in the lung proper. So these are what we're worried about breathing in. Once you get in there, this is what the doctor's worried about trying to get rid of. All right, the challenges in microbiology. We do need a new understanding of the pathogenic mechanisms of bioagents, not only for practical reasons to better develop countermeasure targets and countermeasures, but also because these organisms are very well suited to cause disease, and knowledge about them gives us insight that transcends anthrax or plague or botulism or smallpox, and may be adaptable to other diseases that aren't bioagents but still cause a lot of a lot of misery in people. There's some challenges of medicine with select agents. We need new vaccines. We need new antibiotics. The ones we have are okay for some diseases, especially like anthrax, but they don't work great, and we're always worried about bad people making antibiotic-resistant strains. I believe we need to keep ahead of that. And new antibiotics are important for combating other naturally antibiotic-resistant emerging infectious diseases as well. Medicine is very far behind in coming up with new drugs to treat viral infections. And we need new diagnostics and enough training so people that in the hospitals and clinics and labs can deal if they actually see these diseases. The public health level, we've already, since 2001, the communication between government, different hospitals, and public health people have gotten a lot better. Not every hospital has facilities to handle contagious infectious diseases, the isolation rooms, and we have to make sure that everyone, from first responders, paramedics, and firemen, police to the hospital staff, are fully educated on how to deal with some of these agents. We need new ways to detect these agents. Now, right now, we're really doing it via polymerase chain reaction, which recognizes the specific genetic fingerprints and surface antigens of the bugs via antibody and connecting it to sensors. But we really need real-time ways of detecting these. Now, I know people in Ann Arbor are looking at optics and satellite images and spectra analysis to maybe detect these things from satellites. I'm not quite sure how this was going to work in terms of the large background of natural products that are floating around in the air. But people are working on it. We need ways to sample large volumes of air and give real-time readouts of whether something bad deliberately releases in the air as opposed to the normal things that are found in our environments. The dialogue and vocabulary between scientists, doctors, and the intelligence and law enforcement need to be improved. Right now it's a one-way street. We contribute, but we get nothing back in terms of what's out there, what's the real risk, what should we be focusing on in terms of who's doing bad things in the world. We have to consider some very sticky issues. Are we going to bring back quarantine laws? What are the quarantine laws in Michigan? Are we going to be able to lock people up if they're sick and are at risk to contaminate or to infect other people? Are we going to make people get vaccinated by law? Something we have to deal with is dual-use research policy. Now, there's no primary use. We have no offensive program, so that's not an issue. It's not an ethical issue. But what about information or actual bacteria or viruses that we work with in the lab, or the information we garner from that? Would that information or those viruses ever fall into the wrong hands and be used for bad purposes? So we have to deal with that. And I think as a public, I think this room is an exception what people feel about what germs are and what they can and cannot do. Some people, because they watch some shows on TV, think that if somebody sprays anthrax around, you're going to start convulsing within seconds. It's like a chemical agent. And they don't know that you can get treatment early and that there's vaccines available. Same with smallpox, that you don't have to die immediately. And there's windows of opportunities and things that people can do is interim small steps that'll not make things as bad as they could be or seem to be. All right, that's it. I'll just leave you with this one shot. This is a cutaneous anthrax, looks like a shoulder. And this is the group anthrax. I think these guys look pretty terrifying. Their sounds, some of their basic rhythms they got from the sound of cattle dying from anthrax. All right. And I thank you for your attention.
Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program. Local broadcast is made possible in part by Pfizer.